As you know, proteins are huge macromolecules made up of chains of amino acids. There are only 20 kinds of amino acids, but they can be strung together in any sequence to make up hair, skin, muscles, organs, and of course, enzymes. So the question becomes, where is the instruction book for the amino acid sequence? Right in there, spelled out on DNA. DNA is a macromolecule composed of just 4 kinds of building blocks. We will shortcut their chemical names for now and call them A, T, C, and G. The code for a particular amino acid is a 3-letter code. For example, GAA is one of the DNA words for the amino acid leucine. So the gene for a particular enzyme is a section or sections of DNA carrying the 3-letter DNA words that represent the string of amino acids that will appear in the enzyme. The 3-letter DNA words are like notes on a manuscript page. They represent the exact tune to be played. The instructions for enzymes and other proteins are locked up on DNA in the cell nucleus, but their actual manufacture occurs out in the cytoplasm. So how do the building instructions kept in there get out there? The first electron micrographs of nuclear membrane give a clue. The membrane is festooned with pores. To make a protein, the DNA is opened by an enzyme. Another enzyme, RNA polymerase, charges along one of the strands, the instruction strand, copying the message. It can do this because a DNA building block will only bond with a complementary building block. And on down the line until RNA polymerase hits an encoded termination sequence and drops off. The long molecule of RNA that results is an exact copy of the gene, spelled out in matching building blocks. The 3-letter code for a particular amino acid is called a codon. But this transcription must start at exactly the right place to avoid a disaster. If it started just 1 letter off, all the triplet code words would be wrong, and a garbled protein would be the result. The universal start signal is the AUG codon, and in the protein-making process, it stands out like a green light. The RNA transcript is called messenger RNA, for good reason. It slips out through one of those pores in the nuclear envelope and is picked up by the tiny organelle where the actual translation into an amino acid chain will occur. This 2-part organelle, itself made from a special kind of RNA, is called the ribosome. And cells contain them by the thousands. To make proteins, an animal must have a stockpile of amino acids in its cells, ready for assembly. To get them, it must digest proteins. The amino acids and other building block molecules resulting from digestion are carried from intestine to body cells by the bloodstream. So it's now a matter of lining up an amino acid with its RNA code word, or codon. This happens with the aid of another kind of RNA, transfer RNA, or tRNA. There is a kind of tRNA for each of the 20 kinds of amino acids. The tRNA has 2 bonding sites. One recognizes its particular amino acid. The other contains the anticodon for that amino acid, the same 3-letter code word found on the original instructing strand of the gene. As the ribosome passes along, reading the mRNA instruction tape, matches are found, and amino acids are added to the growing chain. In fact, an assembly line occurs as a number of ribosomes travel along the mRNA, each transcribing the message into identical protein chains. At the finish, the protein will have twisted into its functional shape, ready to do its job, whatever that may be.